come back. So the current waxy corn contains the disrupted waxy gene, and Cativa routinely breed this disrupted waxy gene into elite lines. But however, the backwards procedure takes several years to finish to get the hybrid, and it contains the non-elite genetics as baggage. So the waxing hybrid we got will have the yield penalty compared with the recent elite lines. With CRISPR-Cas assistant breeding, now the Cativa can edit on the waxy gene directly and precisely, and we can reduce the breeding process to 1 generation. So with such advances, numerous research is conducted for CRISPR-Cas in many crops. And if we use CRISPR-Cas with the specific crop name as keywords to search online, we can get the number of publications. We can see in this plot, the most studied crops are rice, maize, wheat, and soybean. And also we can see the publications on rice is around 55. But we can also notice there was only 1 publication for sorghum in 2013. In contrast, sorghum is the 5th most important cereal grain in the world. It's used for grain, biofuel, and forage. We can also see from our everyday life the byproduct of sorghum, such as syrup and beers. So now we know the background that genome editing can speed the crop improvement, and the genome editing platform in sorghum was lagging behind other crops. Our study here is to test a CRISPR-Cas system in sorghum, and also we want to verify the function of 2 candidate genes with this system. So first of all, we need to construct the CRISPR-Cas9 system. We know CRISPR-Cas9 is a combined complex. One of the components is guide RNA. It contains the specifying DNA to cut, and the other component is Cas9 protein. It's responsible for cutting the DNA molecule. And we want to design our system and have this construction expressed in sorghum. So we collaborated with Dr. Bing Yang. Interestingly, he is also the author of the only 1 publication from 2013. And his graduate student, Si Nian, designed the system. So in the first step, we designed 2 guide RNAs for each gene based on the gene sequence and assembled them into our guide RNA scaffold. Next, we will combine this guide RNA vector into another Cas9 vector, and a maize promoter was used here. Next, we will put the whole cassette with Cas9 sequence and guide RNA into an expression vector and transform this expression vector into sorghum. So at last, we will have this DNA segment to express in sorghum, which contains a selective gene bar and Cas9 protein sequence and guide RNA sequence. With this system, we want to test for our 2 candidate genes. These 2 candidate genes are from our lab's previous work. We detected 2 QTLs, 1 for flowering time and 1 for plant height in sorghum. So under the flowering time QTL, we selected 1 gene related to flowering time as candidate gene, and under the plant height QTL, we selected 1 gene related to gibberellin and as candidate gene, and we want to figure out whether GA plays a role in sorghum plant height development. So now I'll tell you the result for the two candidate gene individually. So first I'll show you the result for flowering time candidate gene. So here, this panel shows the gene structure of flowering time gene, and our two guide RNAs came from the first exon of this gene. Here I show the sequence result for one event as an example to show our system induced a biallelic mutation for this gene. So this result shows our system works in sorghum and our target gene was edited. So next we want to know how efficient is our system. So we need to do some calculations based on our result. Here I want to mention these thin grasses. They are sorghum seedlings. So I use them to represent the plants we got after transformation. So after transformation, we got 21 events in total, and there was 37 plants. Of these 21 events, 3 of them are transformed successfully. So that means our transformation frequency is 13%. And of these 3 events, one of them was edited successfully. So that means our editing frequency was 33%. And we developed T1 generation from T0 generation. And we can observe the Mendelian segregation ratio in T1 generation. For the second event, there was no mutants in T0 generation, but we observed novel edits in T1 generation. Then we transplanted 121 plants in greenhouse. Then I babysitted them from a seedling to taller than me. And I extracted tissues to genotype, measured plant height, and bagged self-pollinated them to get the seed for T2 generation. So here shows the result for the flowering time candidate gene. So in this picture, the left plant is the wild type plant, and the right one was edited for the flowering time candidate gene. So we can see from this picture clearly the edited plant flowered has delayed flowering time compared with this wild type. And in the bar chart, we can also see from the statistic test there was significant difference between the flowering time of these two of wild type and edited plants. Then we repeated this experiment in T2 generation, and we got the same result. The edited plants flowered later than wild type. So this result show first this edited gene was able to pass on from T1 generation to T2 generation. Also, this edited gene delayed flowering time in sorghum. Now I'll show you the result for the plant height candidate gene. Similarly, this A panel shows the gene structure of the plant height candidate gene, which is related to GA. And also, these two guide RNAs are from the first exon of this gene. And for this candidate gene, the deletion was here was a big chunk of deletion is caused by these two guide RNA working together. And for the efficiency of the system for this gene, After transformation, we got 15 events in total, and there were only 2 events were transformed successfully. So our transformation frequency was 13.3% for this candidate gene. And there were 5 plants from these 2 events, and 5 of them were edited successfully. So our editing efficiency was 83%. Here in T1 generation, we didn't observe any homozygous mutant. So one hypothesis is the deletion in this gene might have lethal effect. For the phenotype result of this candidate gene, we didn't observe significant difference for the plant height between wild type and edited plants. This is because during our research was conducting, the other 2 groups published their report, and they identified another gene, different gene, underlying this DW1QTL. So it turns out that we target for a wrong gene in plant height. And gibberellin might not play a role in sorghum plant height development. So in summary, we firstly construct our CRISPR-Cas9 system and transform it into sorghum. Next, we genotype them to identify which ones are mutant, which ones are wild type, and combined with the phenotype, we can verify our candidate genes. And as a result, our results show the designed CRISPR-Cas9 system works well in sorghum and is very efficient. The transformation efficiency can be over 13%, and editing efficiency can be as high as 80%. Also, for the 2 candidate genes, we confirmed one gene delayed flowering time, and for the other gene, we confirmed it's not functioning in sorghum plant height. For the future work, our designed CRISPR-Cas9 system can benefit other sorghum studies, and we are positive to apply this system for my lab mate Cheese sorghum gene cloning studies. With that, I want to thank all my lab members, and especially Dr. Yu and Xianran and Cheese help in this project. And Greg helped me a lot in greenhouse. And also many thanks to our collaborators, Dr. Bing Yang and his graduate student Si Nian. They designed the whole system. And Dr. Zhan Yuan Zhang, he is responsible for the transformation process. And our findings come from NIFA, USDA, and Plant Science Institute. And I get the scholarship from our government to study here. And with that, I want to thanks for your attention, and I'm happy for any questions.
Questions for Jialu?
Hi, Dr. Beavis.
Are there orthologs in maize?
Yes.
And for the flowering?
Yes.
And has anybody tried to edit those?
Okay, Dr. Beavis is asking, is there any homolog of this gene in maize? And yes, for this gene, there are homologs in maize and rice and other species, and we are the first one to confirm this gene in sorghum. And I didn't find other studies in maize for this gene. Yes, thank you.
I have a question. For the phenotype of the delayed flowering,
Yes.
as for SBFT, it was a fairly small amount of delay. Is that the expected expected amount based on maybe the previous QTL studies where they looked at the effect of that QTL? Was it about the same amount or was it significantly different? Could you comment on that?
Okay, Dr. Barb is asking about the small difference in T2 generation of the flowering time between wild type and mutant. Yes, the difference of T2 generation is from 1 day to 2 days, but it's still statistically significant. And that small difference might because for our T2 generation, we only use one event, and it's very consistent for wild type, both wild type and the mutants.
So that
yes.
difference was statistical still? Yeah.
Yes.
Okay.
But is it the same as QTL studies?
So there's multiple flowering time genes in sorghum, right?
Yes, yes.
Is SBFT like one of the bigger ones or smaller ones relative to the other genes? Or other QTL?
Okay, the FT QTL is not the largest
Okay.
effect QTL for the flowering time.
Okay.
Yeah.
Can I ask another question?
Yes.
So the first for SBFT, the genome edit or the editing that you guys did produced a deletion of 1 base pair, and the
Right.
second was 4 base pairs.
Right.
The second event, and for the plant height QTL or gene, it was a 21 base pair deletion.
Oh.
Is that expected or?
Okay, Dr. Barb is asking about the deletion, the difference between the two candidate genes. One is only 1 base pair, 4 base pair. The other one is actually 43 base pairs deletion. It happens randomly for CRISPR-Cas9 system.
Okay.
Yeah. Okay, Matt.
For these 2 genes, were they in this? I can't remember now if they were in the same construct in CRISPR, or were they in different?
Okay, Matt's question is asking for do we target for 2 genes at the same time in one construct? So no, we designed. The answer is no, we designed separately for 2 genes.
They were separate.
Yeah, and for each gene, we use 2 guide RNAs to increase the editing efficiency.
Okay. That eliminates my second question.
Yeah. Okay. Thank you.
Question when you do gene editing,
Uh-huh.
is then the T0 or the test edit, is this heterozygote or are both alleles changed?
Oh, okay. Her question is asking about the mutants in T0 generation. Are they both heterozygous or homozygous? So for the flowering time candidate gene, we have a biallelic mutant. So we have 2 different alleles. They are both edited. Yes, they are biallelic mutants, and for plant height candidate gene, there was only 1 allele mutation, the 33 base pair deletion.
But wouldn't it then be possible for the plant type that the not edited allele just makes your plant split?
Yes, it can happen. Excuse me. Sorry.
You're okay. Should we ask your question again, maybe?
Yes. Her question.
If you say for the plant type edit that only 1 allele was edited and the other one is wild type, could it be that you find no effect that is due to the fact that there's still wild type allele present?
Yeah, I think her question is asking there's no difference for plant height. Is that because that's heterozygous? Right. Yes. Okay.
Do you want to call in a friend? Is that what we're doing here now?
It is true that
Yes.
sure you have the ratio because you only have one individual, right? So it might not
Yes,
be
we discussed
statistics, yeah.
discussed it on Monday.
Yeah. That 1 versus 20.
Right.
So she's asking, you did not detect difference. Was that because you have essentially 1 heterologous versus 20 that wild type? That could be different too, right?
Yeah. Okay. Yes. Okay. So, in our analysis, so here I only show the genotype of 21 plants, and one of them are the heterozygous with 33 base pairs deletion. But in our analysis, we actually has other mutant genotypes, and so they come together. The result is it's not significant difference. There's no significant difference.
With this, did you self the heterozygote and see if you could recover homozygous for the mutation, or did you find that to be kind of lethal?
Okay. So his question is asking if we selfing the heterozygous to get the next generation to see if there are homozygous mutants. So we actually did that from T0 generation to T1 generation. In T0 generation, there's 1 heterozygous. So we're selfing them, and there's no homozygous mutant. Yeah. And we also selfing T1 generation to T2 generation, and the germination rate is very high, and there's no homozygous mutant. Yeah, that also proves maybe the lethal effect of the double deletion, biallelic deletion.
So I always hear about off-target
Yeah.
parts of the genome. Did you assay for off-target?
Okay, Dr. Beavis is asking about the off-target issue for CRISPR-Cas9 system. In our study, we didn't sequence the whole genome to see if there were off-targets. Yeah, so we didn't focus on that.
Did you see any phenotypes in the plants that you evaluated that were off target?
No, we didn't. Yeah.
Any more questions? Please join me in thanking Jialu Wei for her.
Thank you.